Hi, I'm David, and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. In question 1, we have an electron photomicrograph of a plant cell. A1 wants you to state the evidence from the figure that shows that the cell is from the stem and not from the mesophyll of a leaf. Mesophyll cells specialize for photosynthesis. They should contain many chloroplasts, which cannot be seen in this cell. A typical mesophyll cell should also contain a large central vacuole to push the chloroplast to the edge of the cell, maximizing the absorption of light. This cell contains a few small ones instead. Due to the presence of a large vacuole, the nucleus of a mesophyll cell is located at the edge or the periphery rather than in the center. 2 wants you to complete the table by identifying the cell structures using their functions. A cell surface membrane allows the diffusion of molecules for gas exchange between the cell and its surroundings. It is A in the figure. The mitochondrial membrane can also be an answer as it allows the diffusion of gas molecules for aerobic respiration. Ribosome synthesis occurs in the nucleus. Cell surface membranes have carrier proteins which actively transport ions in or out of the cells. However, other membranes including tonoplasts, mitochondrial membranes, and the nuclear envelope can also carry out active transport to take in or remove substances according to the needs. The mitochondrion is the site of aerobic respiration where ATP is synthesized. In B, we have a figure showing the movement of vesicles within a plant cell during the development of a vacuole. 1. Name the process that is occurring at X. X shows the cell taking in some substances from the liquid in the surroundings. You can use the term endocytosis or pinocytosis. Note that this is not a process that involves receptor on the cell surface membrane binding with an antigen. So, this is not phagocytosis. 2. Explain how proteins that have not folded correctly are broken down in the vacuole. To break down biological molecules such as proteins, we need to carry out hydrolysis. It means that water is used to break bonds in a molecule. The enzymes that catalyze this reaction are called the proteases. You can also call them peptidases or proteolytic enzymes. They are the enzyme that catalyze proteolysis, a process where proteins are broken down into smaller polypeptides or single amino acids. Peptide bonds are broken in the protein, and amino acids are formed. C. Describe the role of lysosomes in animal cells in defense against pathogens. Notice that the question is about defense against pathogens. Answers related to the digestion of all organelles and the function of phagocytes in the immune response is irrelevant. After the pathogen is engulfed by the cell, lysosomes fuse with phagosomes to form phagolysosomes. This leads to the breakdown or digestion of the pathogen. Then, name 2 enzymes and the substrates they hydrolyze to get another 2 marks. For example, carbohydrates catalyzes the breakdown of polysaccharides in the pathogen. Lipase catalyzes the hydrolysis of lipids. These molecules are digested or broken down to form harmless and soluble products, which may be useful for the cell. In question 2, we have the diagrams of glycogen and cellulose. A wants you to describe 3 ways they are different from each other. Notice that you can only describe the visible differences from the figure. So, do not use your knowledge about them to make statements which cannot be proven by the pictures. The first difference is that glycogen is branched, but cellulose is unbranched. They are made up of different monomers too. For glycogen, it is alpha glucose, while for cellulose, it is beta glucose. You can tell from the picture by the way they form bonds with each other. Alpha glucose do not need to rotate for bond formation as the OH group on its carbon number 1 is on the same plane with the OH group on the carbon number 4. Glycogen has both 1,4 and 1,6 glycosidic bond. 1,6 linkage is found where it branches. Cellulose only has 1,4 glycosidic bonds as it is unbranched. You must state that the bonds are glycosidic bonds. Numbering alone will not be credited. In cellulose, every alternate glucose residue rotated 180 degrees. This cannot be seen in glycogen. B shows the branch structure of glycogen. Explain how the structure of glycogen is related to its function in cells. Glycogen is made up of many alpha glucose, so it is a store of energy or glucose. It is highly branched. These branching points or terminals enable easy addition and release of glucose. In other words, it is easy for our cells to hydrolyze it to get glucose or perform condensation to store glucose according to their needs. It is a compact molecule, so it stores much glucose or energy in a small space. Glycogen is insoluble in water. This is because most of the polar OH groups in glycogen have formed hydrogen bonds with each other, leaving few groups available to interact with water. Storing them does not lower or change the water potential of cells. You can also say that it has no osmotic effect. This is an important feature of storage molecules as we do not want them to affect the movement of water across the cell surface membrane. There is a mark for AVP. For example, joining glucose molecule to form this polysaccharide can prevent the loss of glucose from the cell, as they can't move out of the cell via protein transporter anymore. Glycogen is also chemically inactive, so it does not take part in cell metabolism. A non-reactive molecule is a better storage molecule, as it will not affect the chemical reactions in a cell. C. Explain how the arrangement of cellulose molecules in plant cell walls is related to their function. Cellulose is unbranched and linear. Cellulose molecules are arranged in parallel to each other in the cell wall. Because of this, a large number of OH groups are projecting in all directions. The OH groups allow hydrogen bonds to form with water, making it hydrophilic. They also allow the formation of many hydrogen bonds with cellulose molecules, forming microfibrils. Hydrogen bonds give high tensile strength to microfibrils. This also means that the cellulose fibers formed by the microfibrils and the cell wall formed by these fibers have a high tensile strength. Tensile strength is the maximum stress that a material can withstand while being stretched or pulled before breaking. Due to the high tensile strength, cell walls can resist turgor pressure when water enters the cell by osmosis. So, plant cells do not lyse when they are placed in a solution with a higher water potential. Question 3 talks about membrane proteins called hexose transporters, which can be found on the cell surface membrane of grape cells. A wants you to state why membrane proteins are required for the movement of molecules, such as glucose, across cell surface membranes into cells. In this kind of question, you have to talk about the features of both the molecules and the cell surface membranes. These molecules are polar, water-soluble, and hydrophilic. Saying that they are too large will not get you the mark. Many substances that cannot pass through the cell surface membrane, such as ions, are actually very small. The hydrophilic molecules cannot pass through the phospholipid bilayer or the hydrophobic core in the cell surface membranes. Referring to the fatty acid tails without mentioning the bilayer or hydrophobic core would not get you this mark. We have some information about a hexose transporter known as VVHT1. An investigation was conducted to determine the rate of uptake of glucose by yeast cells in different concentrations of glucose solution. B1. Explain how the results in figure 3.2 provide the evidence to support that VVHT1 is responsible for the facilitated diffusion of glucose into the cell. We can tell from the fact that the rate of uptake becomes constant at high concentrations of glucose. If it is active transport, the rate of uptake should not be affected by the concentration of glucose. The rate of uptake levels off because protein carriers are working at their highest rate. You can also say that they are saturated. The term limiting factor and Vmax can also be used to explain this. But be careful, if you answer by using Vmax, do not mention enzymes, as we are talking about the Vmax of a protein transporter here. 2. Explain why increasing the number of hexose transporters could be commercially important to growers of grapevines. You must refer to the benefit of this to the growers. Do not only discuss the features and characteristics of the grapes. If the uptake of glucose increases, the grapes will become sweeter. Sugar, such as glucose, are solutes. They have osmotic effects. The concentration of sugars in grapes also determine the uptake of water by osmosis, making the fruit more juicy and fleshy. More glucose also means that there are more respiratory substrate in the cell. More energy would be available for the growth of the grapes. This leads to a larger yield. As a result, there will be an increased profit or income for the farmers. There is a mark for AVP. For example, farmers can produce grapes in a shorter time due to the higher growth rate. The growers can also sell their produces at a higher price due to the good quality. C shows us a diagram of a protein used for antigen presentation. 1. State what is meant by a non-self antigen. It is a foreign protein or glycoprotein that stimulates an immune response. You can also say that it stimulates the production of antibodies or the activation of lymphocytes. Stating that it is a polysaccharide, a molecule, or a foreign substance is also acceptable, as protein is not the only biological molecule that can act as antigen. With reference to figure 3.3, explain how T lymphocytes respond to infection by a specific type of pathogen. Your answer should focus on the T lymphocytes, not the whole immune response. And don't forget that you must talk about both the helper and the killer T cells. There are many clones of T lymphocytes in our body. Only the clone of specific T lymphocytes that have receptors complementary to the antigen will be activated. These specific T lymphocytes bind to the antigen on the surface of the antigen-presenting cell. T lymphocytes can only recognize and respond to antigens that has been processed and presented by APC, such as macrophages. The activated T cells then divide many times by mitosis. This is called clonal expansion. The activated T helper cells will then secrete cytokines. You can also call them interleukins or interferon. A detail of the effect of cytokines will give you the next mark. For example, it activates B lymphocytes and enhances the function of macrophages. The selected T killer cells bind or attach to infected cells and destroy them. Any detail of their actions will give you the next mark. For example, you can name the substance they secrete to kill the infected cells. Lastly, there will be the production of memory T cells. Both T helper and T killer memory cells are produced. Question 4a asks you to explain the meaning of the term gene. A gene is defined as the sequence of nucleotides or bases that is a part of a DNA which codes for a polypeptide. You can also use the words proteins or amino acid chain. B shows a diagram of DNA replication. 1. Describe the roles of DNA polymerase and DNA ligase in the replication of DNA. DNA polymerase catalyzes the addition of activated or phosphorylated nucleotides. You will not get this mark if you do not use the word activated or phosphorylated. These nucleotides have formed complementary base pairs with the template strains. Note that the hydrogen bonds between the complementary bases form without the help of DNA polymerase. The enzyme catalyzes the formation of phosphodiester bonds between the adjacent nucleotides. This elongates the strain. DNA polymerase also has proofreading ability. It can fix the mispaired bases in the process. DNA ligase joins the Okazaki fragments together. It occurs on the lagging strand. This is because DNA polymerase can only form a new strand from the 5' to 3' direction. At the lagging strand, as the DNA molecule unwinds a little bit more, some free activated nucleotides will be joined together and form a short fragment. It catalyzes the formation of phosphodiester bonds between the fragments to complete the phosphate sugar backbone. The nicks or gaps between the fragments are sealed. 2. DNA replication occurs during the S phase. S stands for synthesis, which refers to the synthesis of new DNA molecules. In C, we have a diagram of a chromosome. A is the centromere. Kinetochore is not accepted as a correct answer. It is a protein complex associated with the centromere to which the microtubules of the spindle attach. It is not shown in the diagram. A centromere is a site of attachment of chromatids to spindle fibers. The shortening of the microtubules in anaphase causes the separation of the sister chromatids. A centromere also holds the sister chromatids together in a chromosome. B is the spindle fibers. It is made up of microtubules, so you can use this as an answer as well. During metaphase, they orientate chromosomes at the spindle equator. At anaphase, they cause the separation of chromatids due to their shortening or contraction. They pull the centromere and cause the movement of chromatids to the opposite poles. C2 wants you to show what happens to chromosome 11 in anaphase. Remember that the chromatids have separated by this stage. So, there will be two single-stranded chromatids or chromosomes. Do show the attachment of the chromatids to the spindle fibers that are connected to the centrioles. The centromere is leading the movement, so you have to show that they are pointing towards the poles. Figure 5.1 shows a capillary in muscle tissue. A wants you to state the evidence that identifies the cells inside the capillary as red blood cells. Unlike those cells in the surroundings which have prominent nuclei, the cell inside have no nucleus. This is a feature of mature red blood cells to have a greater capacity to transport oxygen. There are no organelles seen in those cells as well. This is also a feature that increases the space available in red blood cells for hemoglobin. You can see that some of the cells are biconcave shaped. You can also say that the cells have different shapes. This is due to the flexibility of red blood cells, which enable them to change shape to fit into small capillaries more easily. Red blood cells have the same width as the lumen of the capillary. The diameter is about 6 to 7 micrometer. This is to allow them to squeeze into the small blood vessels for gas exchange. B. Explain how the structure of the capillary wall is related to its function. Unlike arteries and veins, which have 3 layers of tissue, the wall of a capillary is only 1 cell thick. The thickness is only 1 to 2 micrometer. The wall is made out of endothelial cells. You can also use the term endothelium if you are referring to the tissue. This allows gas exchange to occur more effectively as there is only a short distance for diffusion. The rate of diffusion increases as the distance decreases. There are pores within the endothelial cells called fenestrations. There are also pores or gaps between the endothelial cells of the wall. These pores allow easy passage of small molecules from plasma to tissue fluid and vice versa. Molecules do not have to pass through the cell surface membrane of endothelial cells. Hence, the efficiency increases. Endothelial cells can also perform pinocytosis to take up substances in blood plasma. Figure 5.2 shows us the event that occur during the transportation of carbon dioxide in red blood cells. 1. Explain how an increase in carbon dioxide in the blood leads to the release of more oxygen from red blood cells. You should discuss the events when there is more carbon dioxide. Do not only describe the sequence of events without referring to this change. A higher partial pressure of carbon dioxide leads to more carbon dioxide diffusing into red blood cells. An enzyme in red blood cells called carbonic anhydrase catalyzes the formation of more carbonic acid. It will then dissociate to form more hydrogen ions. Hemoglobin has a higher affinity for hydrogen ions compared to oxygen, so it will bind with more hydrogen ions to form hemoglobinic acid. This decreases the affinity of hemoglobin for oxygen, leading to the release of more oxygen. There is a second event that also causes the release of more oxygen. Carbon dioxide can bind to the N-terminal of globin. Globin is the name of the polypeptides that form hemoglobin. Carbaminohemoglobin will be formed. This lowers the affinity of hemoglobin to oxygen due to the allosteric effect. The allosteric effect refers to the change in the tertiary structure of hemoglobin. This change in shape favoring the dissociation of oxygen. C2. Explain why the concentration of chloride ions in the blood plasma of deoxygenated blood is lower than in the blood plasma of oxygenated blood. This is due to the chloride shift. In the diagram we have on the previous page, hydrogen carbonate ions move out of red blood cells following the dissociation of carbonic acid. Chloride ions in the plasma will pass into red blood cells to replace the negatively charged ions. This is important because we do not want the movement of hydrogen carbonate ions to cause an imbalance to the charge across the cell surface membrane. Chloride shift helps to achieve electron neutrality, which ensures the stability of the membrane. There is a mark for AVP. For example, you can state the method chloride ions pass through the cell surface membrane. Question 6 shows the passage of water through the tissue of a flowering plant from the soil to the atmosphere. X is a channel that links the cytoplasm of adjacent cells. It is a plasmodesma. The plural form is not accepted because only one structure is labeled. Y is found in the cell wall and is a barrier to the apoplast pathway. It is the Casparian strip. It is made up of suberin, a waterproofing waxy substance. Due to its hydrophobic nature, water is not allowed to pass through the cell walls of endodermal cells. When water moves from the soil solution to the cytoplasm of root hair cells, it moves down the water potential gradient and passes through the cell surface membrane. So, it is osmosis. The upward movement of water in the xylem is due to the transpiration pull. The loss of water from the leaves is the driving force for this tension. Besides, hydrogen bonds between water molecules cause them to move up together as a continuous column. This is called cohesion. When water moves from mesophyll cell walls to intercellular airspace, it changes from its liquid to gas state. Water turns into vapor by evaporation. Water vapor will then move to the atmosphere outside the leaf by diffusion down the water potential gradient. That's all for today. If you think my videos are useful, don't forget to like, comment and subscribe. Thank you for watching and see you again soon.